Hi everyone, and welcome to our video on discontinuous and continuous variation. This continues our Module 6 Part 2 series of videos looking at those patterns of inheritance, and in this video we'll be having a look at the specification reference 6.1.2d, the genetic basis of continuous and discontinuous variation. And you can see that you should have reference to the number of genes that influence each type of variation. It's one of the more common multiple choice questions they like to ask you about this topic. Okay, so we're going to start off with discontinuous variation. So when we're talking about discontinuous variation, this is where we've got genetic variation producing these discrete phenotypes. And what we mean by that is that we've got two or more non-overlapping categories. So basically the phenotypes that we're getting are discrete groups. And I've given you two common examples there. So sex, male or female, if we're talking humans. And then blood type, we've got A, B, AB, or O. There's no little bit of this, little bit of that. It's either one of those four types. So it's that straightforward in terms of discontinuous. You must have a discrete phenotype and they don't overlap in any way. So they are very much unique little entities. Now, when we think about that genetic basis of our variation, this discontinuous variation is going to be controlled usually in monogenic ways. Now, if we break down that word monogenic, mono meaning one and genic obviously referring to a gene. So what we're seeing here is that this type of variation, discontinuous variation, will have a single gene locus or maybe two genes that are involved in actually controlling it. So literally one or two gene loci that will therefore be involved in controlling this, and we're going to end up with these discrete phenotypes. So if we consider what being monogenic actually means, then where we've only got that single gene locus, if we have a different allele present at that one gene locus that's controlling the overall phenotype, then clearly we're going to have a significantly large effect on it. So it can completely change that phenotype. And if we think about our blood groups, then obviously you've got your A, your B, and having just that different allele present can change you from the potential of type O blood to type A blood. So different alleles at that single gene locus have a large effect on that final phenotype. But as I've mentioned in my previous little moment, we have got those scenarios where there are potentially two gene loci involved in this. So as much as most of them are monogenic, sometimes we do have two gene loci involved, but in those scenarios, they will be interacting. And the interaction between those two gene loci is going to influence that one characteristic. And one of the best examples that I can give you of this that you've done very recently, epistasis. So we had a look at those little mice, for example, at the end of our epistasis video, where we have got those two different gene loci involved. Obviously the one that's going to actually change that colourless precursor into a coloured pigment, and the other one that determines the distribution of it. So we've got those two gene loci involved. They're influencing the one characteristic of the coat colour, and we're ending up with three discrete categories, three discrete coat colours. They're either going to be albino, black, or agouti at the end. Now, it is worth remembering when we are talking about discontinuous variation, this is based solidly in the genetics. So the environment isn't going to be playing a role here. It is about the alleles that are present. So discontinuous variation, one or two genes are going to be involved in this, and there's not going to be an influence from the environment. The second type of variation that we can talk about is the continuous variation then. Now, continuous variation is one that's going to produce phenotypic variation where quantitative traits vary by very small amounts. Now, basically what that means is we've got a whole range of potential values that we could take between some extremes, okay? So it's not like there's an infinite number of possibilities here. There are obviously a maximum and a minimum value for anything that we're talking about, but between those maximum and minimum values, we could have a huge number of potential intermediates because they just kind of flow from one to the next really. And I've given you some examples here where you can get that idea. So height, it's not a case of you are either 5 foot or 6 foot. You are obviously maybe 5 foot 5.5. So there's a whole range of little intermediate values that we get there. Skin color, again, you're not black or white. There's a whole range of different shades of skin color that you can get between those as well. So what we find is that there are these little differences between the individuals, and that gives us this continuous range of values that we could have. Same kind of idea with the eye color there. And then let's move away from humans for a moment and go into some other organisms, because it's not just humans we study in this course. The length of leaves, again, they will have a range of values they could take within obviously given range. And then the length of tails of creatures as well. Maximum and minimum sizes, of course, but the actual length of individuals' tails will take a range of values within there. So when we're thinking continuous variation, we're looking at very small variations from one group to the next. In terms of what actually controls this continuous variation then, we don't just have 1 or 2 genes, we've got many genes. And so because we've got many genes in determining this final characteristic, we refer to them as polygenic. So remember, we can break that word down. Poly just means many and genic, genes. So we've got many genes actually involved in this final determination of our characteristic. Because we've got many genes involved, then what that means is each gene can contribute a small amount to the phenotype. So what we're really seeing is this additive effect. So you get obviously one gene with a particular allele present, and then the next gene that has an influence, that allele will have a little difference, and so on. So what we actually find is that the more gene loci we have that contribute to a given characteristic, the more continuous the variation becomes, because each of those individual gene loci that contributes gives that little difference. So if we've got lots and lots of gene loci, they're all giving very small differences, and therefore when we actually look at the variation that we end up with, it becomes more continuous. There's just smaller differences from one category to the next. Now, unlike our discontinuous variation where the environment did not have an effect, when we're talking continuous variation, the environment does have an effect, and it can have a significant effect, in fact. If we go back to our example of height that we looked at earlier on, it doesn't really matter whether you come from a family of 6'6" giants if you've got a terrible diet in your formative years. So if you are not getting a good diet with lots of protein to actually give you those building blocks to let you grow to your potential height, you're never going to reach it. So as much as you might have all of these genes that mean you could be another 6'6" giant person, then if you don't get the diet, so your environment isn't giving you those things that you require to get to that potential, you don't reach it. So do bear in mind that when we're talking about this continuous variation, the environment does have a significant part to play. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when more videos are uploaded, and head on over to the website where you can find a range of resources to help you in your A-level biology studies.